So today I'm going to speak a little bit about tRNA as well as some of the mutations that are usually seen in our normal day-to-day curriculum of biochemistry and genetics. However, this is in fact a bit this includes some of the biochemical stuff as well as certain genetics which are sophisticated and require a little bit of attention. However, I will just like to bring it down to a level which is very elementary and could be understood by almost anybody. But we'll try and see some of the major impacts that is carried out by how the mechanics works as well as how the mutations goes through its effects. Right. Before starting anything, I just would like to express some of the important features that are found in the genetic code. It's this is quite trivial, but still, I would think that this has a better point to start or initiate my lesson. So the genetic code, the first thing, first criteria that I would like to discuss is the degeneracy. Now, if you think there are, even though the specificity of genetic coding is there, what usually you see is that most of the time there are around 60 codons. So most of the time you would see that a single amino acid will have multiple codons. How is this possible? We'll be looking into this in our as the lesson progress, but it is something that you should know that for the single amino acid there are multiple codons that are present. But it is also, there is another criteria that we uh, would like to discuss along with this, is the uni-ambiguous nature of the genetic codon. Now, what it says is that now here in the degeneracy, what you would see is multiple codons for amino acids. In the uniambiguous nature, what you would see is that a given codon will always represent a single amino acid. Now, that's a criteria that can be related with the specificity that you see in the genetic coding. But it is something like this. Now, this degeneracy is brought about by the action of the tRNA molecule itself. The tRNA has a structure which is known as the anticodon that is involved in bringing of or activating a certain amino acid so that a polypeptide chain is produced. So this degeneracy has something to do with tRNA. I'll explain it as we proceed. So these are two features that I would like to bring at the initial state of our lesson: the degeneracy of the genetic code and the uniambiguity. Right. What are the other features? Just for the sake of completion, there is something called non-overlapping, which means now if there's an mRNA with codons, the codons will already, all the codons that are present within a mRNA molecule will be read, and to that a complementary amino acid will be brought about as a protein. However, there will never be overlapping of these codons, and some might not, and there is no occasions where any codons will be either not read or not taken into account. So non-overlapping is a primary feature that you see in the genetic code. The fourth would be no punctuations. Now again, this also plays an important part in mutations. Now even now in between codons, let's assume this is codon number 1, this is 2, this is 3, this is 4, and there is no punctuation that is first present in between them. So if by any circumstances, if a single nucleotide becomes deranged or undergo mutations or inserted or deleted, this during the process of translation, you will never be able to identify that. Therefore, there is a possibility the mutations could be carried out. Now, there's a specific thing called frameshift mutations, which are due because of the fact that there is no punctuation. So again, important criteria that you see in the genetic code. Now, for degeneracy, uniambiguity, non-overlapping, and no punctuations. The final would be the universal nature. Now, because of the universal nature, we have the capacity to produce certain molecules in a very in a more industrial manner. For an example, insulin, erythropoietin are proteins that are produced through recombinant technology. This is present because now the genetic code for translation purposes in a prokaryote is similar to that of a eukaryote. Therefore, plasmids can be utilized in a similar manner via recombinant DNA technology to produce our own genes, to produce our own proteins. That's why this plays an important role in the new technology, the new DNA recombinant technology. Yes. So these are the 5 features that you see in the genetic code. Now, I'm just going to dive deeper into degeneration and uniambiguity because now my topic would be rather the tRNA molecule itself. Right. Now, as I said, this tRNA molecule has a 3D structure. It has a few loops. There's a D loop, there's a T loop, right? The D loop is involved in, it usually binds to a certain enzyme which is known as amino acyl transferase, and the T loop has the ability to bind to the ribosome. Again, an important location because the translation takes place at this specific unit. And then there's the anticodon loop. Now, what does the anticodon loop do? Here's the anticodon. Now, it binds with the mRNA molecule in a more or less a complementary manner so that it will cause the activation or the bring about of a specific amino acid which is required in the production of the polypeptide chain. Are we good? Yes. So these are the three. I'm just going into the structure. Now, the question arises, how does the tRNA molecule identify a specific amino acid according to the codon that is given? Yes, it is because the anticodon will bind to the codon in a complementary manner. But the anticodon doesn't have direct relationships with the protein or the amino acid. So the amino acid, which in fact binds to the adenosine group that you see in the third 3' location, and this amino acid this amino acid to be brought from an amino acid pool in a very specific manner requires the action of an enzyme which is known as the amino acyl transferase, which acts as the messenger. So simply what it does is that it acts as the messenger and then brings the specific amino acid to involved in the translation process. So are we good? This is an energy requiring process. Now in the Harper, the it's a biochemistry textbook. The Harper shares about this is brought about very clearly how the tRNA molecules is activated. It is activated by means of binding to an adenosine monophosphate group, and with that it involves in the translation, and later the adenosine monophosphate is released after the translation is complete. Now the thing that I need to bring about is that at least there is a single specific tRNA for an amino acid. At least one. Now there could be more, but at least there's one of them. Even though I have uh, spoke about the structure of the tRNA molecule, I have not exactly said how the degeneracy works. Now it's something like this. Now for an example, if I take a tRNA molecule back again, there is this, the accept arm, then there's the D loop, the anticodon, and the T loop. Right. Now, what does the anticodon do? The anticodon will bind complementary to the codon. I'm going to illustrate this by using a simple example. For an example, as an example, I would like to take the codon which codes for arginine. The codon would be AGA. So AGA, the anticodon that is found for AGA is A should be bound with uracil, G with C, cytosine, and A with back again uracil. So this is what you would see. But now once this anticodon, the specific anticodon UCU binds to this, what would happen is that it will cause the tRNA molecule to be activated by the cofactor which is known as adenosine monophosphate. The adenosine monophosphate, with the binding of the adenosine monophosphate, it will cause the binding of a specific amino acid transferase, T amino acid transferase, which will act as a messenger to bring about the specific molecule amino acid which is arginine that will be bound to the acceptor arm of the tRNA molecule, and thereby the translation process proceeds. However, if the codon composed of something like this A, G, G, where the change has only occurred in the third nucleotide of the codon. Now, once this happens, usually what should happen is that the complementary codon, the anticodon for this should be UCC. So as I said, initially this was AGA, which was read by the anticodon UCU, but it's a simple change in the third nucleotide AGG. The third nucleotide should be read by an anticodon which composed of UCC. However, if they are abiding by the Watson and Crick's rules with the complementary base pairing. However, this is not seen in tRNA molecules. They tend to be bit not that not very specific about the third nucleotide. They tend to be a bit mischievous to that. So what the AGG we can bind to UCU as well. It can be bound to by the anticodon, which is in fact specific for AGA. This is known as wobble phenomena because it can wobble about a bit in for the third nucleotide. This is the phenomena that will lead to degeneracy. Now, therefore, both AGA and AGG, both of these codons will code for arginine. So this is how you see the degeneracy because of the tRNA molecule. Are we clear? Right. Now let's proceed to mutations. Of course, this is going to be a bit complicated. I'm just going to give this in a very simplified manner, not going into much details. Now, mutations initially I would like to differentiate or classify them. Classify them. Right. Mutations happens because of changes in the nucleotide sequence. That almost everybody knows. Nucleotide sequence change. If this change happens only at a single point or a single base, this is known as a point mutation. In a point mutation, only a single nucleotide base is altered. Base altered. Right. Now, this can happen by many processes. One would be the transition. Transition is an instance where you would see a change of one purine to another, or one pyramid into another. Again, a very simple process of mutation. Usually this will not be manifested. However, this could again can have its effect if you are very unlucky. Yes. Then the second would be, it is known as transversion, where you would see change of one purine to a pyrimidine. It can either be any of the pyrimidines, or a one pure pyrimidine to any of the purines. Bit more, uh, the effects or the manifestations would be more drastic to that of transition. However, again, a simple point mutation, change of one nucleotide base to another. The difference between transition and transversion is the transversion you can change from purine to a pyrimidine. Bit more, the change is a bit more elaborated than that of a simple transition. Right. Once a mutation occurs, there can be different manifestations. For an example, let's assume that it has underwent a certain mutation at a single nucleotide base. However, the same, now I'm just taking AGA for an example. AG has underwent a point mutation where it has been converted to AGG. Both of these are coding for the same amino acid. So will there be a difference? No. So ultimately you would not see any change or mutations happening or manifested in this individual. Therefore, such mutations are kept silent. Hence the term comes silent mutations. Silent mutations. Are we clear? Silent mutations are correlated with the degeneracy. Now, I hope that you can understand that, because it's very elementary again, because both silent mutations as well as degeneracy happens because of the same molecular factors. Right. Now, the second type would be where a change happens, let's assume a UA or something of that sort. I'm not particular about this. I do not remember the codons and the amino acids that are being related to it. I just say that there is a change, and because of this change, what would happen is that it codes for a different amino acid. Different amino acid. Now, of course, now since there's a different amino acid that is coming into play, there is a possibility that it might show changes or different consequences in the individual, and therefore there will be mutation effects. Right. So different amino acid. Now, According to the location of this amino acid, it can be further classified into three types. The manifestations would change according to the location. They could either be acceptable, partially acceptable, or unacceptable. Or unacceptable means now the changes are very drastic. The changes are quite significant, and there is a possibility that the protein that is produced at the end will not be able to function normally. Hence, unacceptable. Now, this is because most of the time, because of a change of a single nucleotide base, the different amino acid that is being brought about will have the same functional groups to that of the amino acid that should have been in its place if the mutation has not occurred. So, because of the same functional groups and the same functionality of the amino acid, the mutations will not be very drastic. Hence, there is a possibility that it could be acceptable, sometimes even partially acceptable. But there's a possibility it can be unacceptable as well. Right. Now, here I have brought about this, I have taken from the Harper's Illustrated Biochemistry book. It's just that now there is this codon with three A's which has underwent a certain change um, to form either AAU or AAC. Here you would see instead of lysine at the 61 position, you would see asparagine. But the beautiful fact about this protein is that even though once it is been produced by an asparagine, this is in fact the hemoglobin A strand. The hemoglobin A strand will function as it is to that of a normal hemoglobin, will not have any change whatsoever. So this is called the Hikari mutation. The Hikari mutation will not have any changes because asparagine is functionally similar to that of lysine. The only occasion where we can identify is that because of the difference that they have in their molecular weight, this can be electrophoretically distinguished. They will have different masses. Therefore, they can be segregated and identified by these physical measures, but no change chemically or functionally. Then the partially acceptable missense. So such changes where there is this different amino acid replacing the actual amino acid that would have to be there is known as a missense mutation. Now, missense mutations, now as I said, there could be acceptable ones and there could be partially acceptable ones, such as in sickle cell anemia. In sickle cell anemia, glutamate, now see the position, it is present at 6. Glutamate is substituted by valine. Now, the problem that we see here is that it works all right, but this will, because of the valine, now the hemoglobin molecule, the hemoglobin protein that is produced will have a higher affinity towards oxygen. It will function normally in the normal oxygen state. But once the body is succumbed to a more deoxygenated status, what would happen is that the hemoglobin molecule will start to sickle. So the only problem that arises is under circumstances where there is less oxygen or in deoxygenated state. The sickling crisis arises at that particular state. So partially acceptable when it's when in the normal environment. But once the environment is deranged, it will show its manifestation. So partially acceptable missense mutation. The other is the unacceptable ones. Boston. This is the hemoglobin, again hemoglobin A. Histidine replaced by tyrosine at 58 position. You would see that this molecule is known as Boston hemoglobin. The Boston hemoglobin is, once it binds to oxygen, it binds permanently. It will not dissociate. Therefore, the functionality of transportation of oxygen from one place to another, from a higher oxygenated place to a low oxygenated place, will not happen because of this. So it will not function. Hence, this is an unacceptable missense mutation. Such individuals, you should die. So this is how missense mutations can be illustrated. Uh, Easy, but again a bit of a complicated thing. The other mutation that I'm going to illustrate is nonsense mutation. Nonsense, like, what does nonsense means? A point mutation has happened where now it codes for a stop codon. For an example, let's say that there was the fifth codon has undergone mutation where it now code for a stop codon. The actual stop codon is somewhere here. Now, once the mRNA goes on, it will identify this as a stop codon and will stop producing the polypeptide chain. So the protein molecule will be not complete. Hence, it probably will not be functional. So this is problematic and also will show major significant changes in the manifestations in comparison to that of the previously explained mutations. Right. So what are the three types of mutations that we see according to the manifestations? It could be silent, it could be missense, it could be nonsense. Right. The other type of mutation that we come across, now this was only because of a single change of a nucleotide base. The other is frameshift mutation. How does this occur? It is simply because of an insertion or a deletion of a nucleotide base. Now, as I said, according to the features of the genetic code, you see no punctuation. So, for an example, this is, I'm just relating one. This is one codon, and this would be so. Let's assume that there's going to be an insertion of a or a deletion. Let's take a deletion. There's a deletion of a single nucleotide base. This has, now what would happen? Now after this, let's take another one. Right. So the first would be the same amino acid. However, after the particular point where this mutation has happened, there will be a shift of frames. Now there's only two. Two is not acceptable for a tRNA molecule. The tRNA anticodon should bind with three. So what it would take is that it will take a nucleotide base that is present in the consecutive place and code a will involve or bring about a different amino acid into the polypeptide chain, something totally different, and thereby the total uh, genetic code itself will be garbled. It will be completely deranged. it will not show the same, because it's not a single amino acid that's going to be changed. It's the total chain itself is now altered. Now, consequence will be the production of a completely different amino acid chain, which will not show any function whatsoever to that of the normal polypeptide chain that would have been there if such mutations does not occur. So, did not occur. So, this we will see a garbled translation, um, and sometimes even because of such frameshift mutations, there's a possibility that you might come across a stop codon premature to that of the actual stop codon, and there is a possibility that the polypeptide chain will be seized in a more shorter version, so that it will not have its length as well. I know it's a single amino acid that has changed, but still the manifestations are quite drastic because of the shift of frame, right? But this, if deletions occur in triplets or insertions occur in triplets, a codon itself is going to be uh, taken out. Under those circumstances, you would see again there is a possibility that a single amino acid will not be shown in the protein that will be translated, but still the others will not. The distal part of the distal part of the mRNA, uh, which is present which is present distal to that of the mutation, will not have any change. That will be there, or will be showed in the polypeptide chain. So, under such circumstances, the manifestations would not be that drastically significant to that of a single deletion, right? Um, so these are the actions I just wanted to relate about the mutations as well. Now, something very not very important in medicine per se, but still this was expressed in some of the major books that you come across in biotechnology, because these are somewhat now utilized in modern technology for the production of medicine, as well as certain um, uh, research based processes. So I'm just going to relate about what this means. There's a certain entity which is known as suppressor tRNA molecules. Usually, the tRNA works with a very good accuracy. They will their accuracy and a possibility of a compromisation occurring in the tRNA, which will lead to a mutation, is around 1 into 10 to the power 4, which means that the action of tRNA is very accurate. But sometimes, in mostly prokaryotes and low eukaryotes, there are abnormal tRNA, which has the capacity to bind and decode altered genes. These are known as tRNA because of the suppression that is being implied upon the altered genes. These are known as tRNA suppressors. But the problem that is associated with them is that now these abnormal tRNA cannot distinguish mutations from normal genes. Therefore, they will carry out mutations for the normal genes as well. So, in a very awkward fashion. So, the normal genes will be manifested as mutations. So, that is a possibility. usually not seen in higher eukaryotes, but can be isolated for research purposes. Now they are utilized in many research and biotechnological processes nowadays. Just for the sake of knowing, I have said that there is a certain entity such as this. Doesn't need to be confused about it or worry about it. Just know this. Right, we'll move into the questions. Now, this question again in MCQ that I have chosen, it's very tricky as well to get an ideal answer for this, but we'll go through and we'll try to clarify some of the statements that were given. Right, it undergoes post-translational modification. Yes, out of all, it includes tRNA molecules as well. The tRNA should show post-translational molecule modifications for its functionality. If such modifications does not happen, what would there is that there's a possibility it will not function, and also it will show mutations. So for this particular purpose, it should undergo post-translation modifications. It's a true statement. True statement. Yeah. It shows dual specificity. B relates about dual specificity. How is the dual specificity comes into play when it comes to tRNA? It's something like this. Now, the tRNA molecule incorporates both anticodons as well as a specific amino acid transferase enzyme, which brings about a specific amino acid into the play for the formation of the polypeptide chain that will produce a protein. So the dual specificity is by both of these criteria, by one by the amino acid transferase and one by the anticodon. So that thereby it makes sure that the a specific amino acid is brought for the formation of the polypeptide chain. Again, a true statement. Yeah, true statement. Single tRNA molecule recognizes more than one amino acid. Single tRNA molecule. Now, as I said, it shows degeneracy, all right. The tRNA molecule anticodon has a possibility to show wobbling phenomena. So it can identify more than one codons, but it brings about or activates the process in which a tRNA molecule activates an amino acid and brings it about for the formation of polypeptide chain is known as an amino acid charging process. So the amino acid charging process is specific. Now, even though it shows degeneracy and two or more codons can be functionally implied to bring to carry out amino acid charging, but it causes charging of only of a single amino acid, in this case arginine. Both, I know it can read both AGG as well as AGA, but both of these will identify only a single amino acid. That is why it is very specific, and therefore this statement is false. See at the depth, look at the depth that the questions has been posed, and the answers that we have to bring. So these statements says about the tRNA that travels along the mRNA in transcription. Again, a false statement. Why is that? It's because now, if you see, now this is the let's assume this is the mRNA molecule. This is the ribosome. The ribosome is the one that moves from third prime end to the fifth prime end, reading the total mRNA molecule. And as it does so, it incorporates these tRNAs to bring certain amino acids for the protein, for the translation process. It's not the tRNA that moves from one point to another, reading all the mRNA. It does not have that functionality in the translation. It's the ribosome which does it. With the action of translation factors as well as the rRNA which comes into play. That is how the translation process is carried out. It's not the tRNAs. Look at the depth and look at the specificity of these questions. So tRNA travels along the mRNA in transcription is a false statement, completely false. An E statement. Translation is stopped when it binds to a stop codon. Again, you have to read it very in a very discreet manner to see this binds a, a tRNA molecule. The anticodon will there is no anticodon for a stop codon. It will simply uh, cease itself. It will not proceed further. Therefore, there will be no binding as such. Their translation will be stopped by a stop codon, but there will be no binding. Therefore, again, a false statement. So that is how I just wanted to describe this in a very in a very descriptive manner. But see, look at how the questions have been asked and formulated, and to make sure that you get uh, uh, might not get the correct answer. They have tried to play with certain words as well. So make sure that you read the statement in a very clear mind to see the question and to identify and as well as understand what they are trying to eject. So again, now as I have finished, I have just tried to relate some of the structure and the functionality of the tRNA molecules. Along with that, I have tried to describe the mechanics of mutations, which is quite sophisticated and quite mm, uh, elaborative as well. Now this cannot be easily said and done within 30 minutes or so. It just requires a lot more. But uh, the basic phenomena that is underlying behind this lesson is of the structure that I have said. So try to understand this and make sure that you have a general idea about how mutations happens, because this is an important lesson when it comes to medicine as well as its paramedical stuff as well. So make sure that you know this, have an idea and relate it to the other purposes as well. So this is the third video that I have done in genetics. Make sure that you go through my previous ones as well, because I have said about mutations in a very mutations in few other lessons as well. So to get a better understanding, make sure that you go through that as well. So thanks again, and we'll meet next time.